Thanks, Phoebe. So as Phoebe said, I'm going to be talking about reconstructing major functional transitions in the history of life. And really, it's quite a methods-based talk today. So I'm going to talk conceptually about how we try and do this and how we tackle the missing data that inevitably exists when we start looking at fossils and how we can overcome that to try and understand how extinct animals might have stood and moved and interacted with their environment. Um, So I'm actually going to start by talking a little bit about my research group. So I've been at Liverpool for 15 years now, but I was only recently made aware of this seminar series. So I thought it might be useful to actually just introduce the research group that I'm part of in case equally people who come to this series are not aware of our group and what we do. So I'll talk a bit about evolutionary biomechanics and how we do that at Liverpool. And then in terms of coming back to the title of the talk and understanding functional transitions in the history of life and how we reconstruct how fossil animals live, I'm then going to go through three case studies. So I'm going to look at the evolution of body shape in birds and what that tells us about the evolution of locomotion in birds. I'm going to talk about reconstructing muscle evolution in the jaws of rodents and the methods that we use to reconstruct muscles that are not fossilized in extinct animals. And then perhaps most relevant to the audience today, I'm going to talk about running performance in Australopithecus afarensis and some recent work we've been doing on that. So starting with our group at Liverpool, we're quite a big group actually. And although we are an evolution group predominantly in the research that we do, we're actually based in life course and medical sciences, so quite an applied research institute, if you like, and we're very much the odd ones out there. And we work on a range of different animals from humans and human ancestors right back to dinosaurs and fish and various other extinct and extant animal groups. But what brings us together is that we're basically interested in gross anatomy and function. So any aspect of anatomy or form, as I've called it here, that is basically bigger than a muscle fiber. So everything from a muscle fiber right up to the whole body shape of an organism, how different aspects of form or anatomy in that context are related to function, like locomotion or feeding or respiration. And basically what we want to understand are what the functional consequences of evolutionary change in morphology that we see in the fossil record or across diverse living groups, what the functional consequences of those morphological differences and changes are, and then what drove them through deep time. So coming back to this idea of major functional transitions in the history of life. As I say, we are in quite an applied medical group. So what we also do is ask similar questions within humans or companion animals for medical and veterinary research, but predominantly we are an evolution group. We have some very nice facilities for measuring how animals move, measuring their functions. So this is our main gate lab. It's about 18 meters long from that wall to that wall and about 12 meters wide that way. And it's full of very nice kit. So we have a 12 camera motion capture system for 3D motion capture. We have force plates embedded in the floor for measuring ground reaction forces. We have strength testers. We have EMG systems for measuring when muscles switch on and off. And we have instrumented treadmills as well that give us ground reaction forces and plantar pressures at controlled speeds. And the kind of sort of day-to-day data that we might collect in this lab looks something like this. So we'd have a human volunteer covered in spherical markers that are tracked by our motion capture cameras. And from that, we can also measure ground reaction forces from our force plates, and we get body segment motions, as you can see here. And we don't just do this with humans, we do animal work as well. So this is some work we're doing on dogs, looking at selective breeding in dogs and how the wide array of body shapes that we've bred in the domestic dog, how those body shape differences manifest themselves in different motion patterns. So we use medical imaging to quantify body shape and then measure their motions and try and understand the links between the two. We're also very lucky at Liverpool that we have another system for measuring motion. So obviously in the main gate lab, if we're sticking markers to the skin or even to the clothes, that's a very abstract way of measuring how the bones are moving. With this system, which is called a biplanar X-ray system, there's only 3 of these in the UK. We can actually see how the bones are moving directly and then reconstruct their motion with sub-millimeter accuracy. So here's the 2 X-ray planes here, and by sort of triangulating the volume that the arm is moving in, we can then match, we can take 3D models of the bones from the same subject whose arm is moving and animate them according to how they're moving in these videos in 3D. And, you know, it's nice to have that accuracy, but in some cases, like in the human wrist, where you've got all these tiny bones, it's actually the only way to measure how these bones are moving. You can't do it with external based motion capture. We also, as I've kind of hinted at, use a lot of medical imaging. So a lot of MRI and medical and micro CT. This is a day where we moved a 110 kilogram ostrich from our freezer to the small animal teaching hospital at Lea Hurst to CT scan it. That was not a lot of fun. And we do this because it allows us to quantify anatomy in lots of different ways and with relatively high resolution, so that we can then understand how anatomical variation correlates with either the way people or animals move or the amount of energy they use when they walk. So this was a human evolution example. We looked at various features in the limbs and feet of normal, healthy humans to see if the magnitude of expression of certain characters that have been argued to be important in the evolution of human bipedalism, if they correlated with differences in energetics in modern humans. So to put it crudely, if your morphology was more stereotypically modern human-like, more evolved, if you like, do you have a lower cost of transport as a result of having a particular feature that is considered adapted for bipedalism? And what we found is that there was no correlation, actually. With this medical image data and these 3D models, we can also go a step further, and rather than just correlating anatomy and motion, we can actually start to understand the mechanistic links between them. And we do this with biomechanical modeling. So we can take our 3D models of bones, our 3D reconstructions of the muscles and the fiber architecture, and put these into physics-based models where gravity acts on the model. There's basic mathematical models of muscle contraction. All these body segments have masses, so they have to have forces exerted on them to move, just like a real animal. And we can simulate a recorded motion. So here we're recording, we are simulating a recorded motion on a strength testing machine where the individual was pushing and pulling as hard as they could with their ankle, and it was measuring the torque that the ankle joint was exerting on the tester. And then we can simulate that situation and compare how our simulated model compares to the real data. In this instance, it works quite well, within 10% of the real values, which gives us some confidence when we go to simulate more complex situations where it's more difficult to validate. And the reason we're actually modeling is because we can't measure the parameters we're interested in directly. So we can't stick gauges and transducers surgically into healthy human volunteer muscles because it's unethical. But we can use these models to actually make predictions about how the muscles are operating on different substrates, for example, in this example. And it tells us something about why people move differently in different circumstances. We can also go one step further. So in these previous examples, we'd recorded the motion data. You can see the spherical dots here where they were placed on the experimental subject. And the simulator is matching the recorded motions. But what we can also do is try and predict biomechanical performance or task performance without measuring any motions. We can use an optimization approach. So in this particular example, we've got a very similar model here of a human. And we're asking the model to use a machine learning approach to search for the activation pattern that essentially allows the model to jump as high as it can. Oops, wrong direction. So after 1,000 attempts at trying different muscle activation patterns, it's not doing very well. It's kind of starting to do something that resembles a jump, but it's not really jumping very well. If we continue to allow the machine learning to search through more and more different combinations of muscle activation patterns, it starts to find ones that are slightly better. So it is actually getting its feet off the ground at this point. If we go even further and search through more and more patterns, we can ultimately end up with something that looks like a human jumping up on the spot, which is what we were after. Jump as high as you can in that amount of time, and that's what it comes up with. The obvious question with an approach like that is, does it work? And it does seem to work pretty well. So we've simulated walking. Here's the jumping example I just showed, and running as well. And when we compare the model's predictions to, walk as efficiently as you can, jump as high as you can, run as fast as you can, we get quite good results. So the simulator is in blue here across a range of walking speeds. And then we've got the energetic cost measured in that subject with the green dots here. And you can see it matches really well. In terms of jump heights, the maximum jump heights that I showed in the previous animations are within 10% of the values measured for that human subject. And something I'll come on to at the end when I talk about running in Australopithecus afarensis is the simulator, which is the data points here, predicts cost of transport that overlaps the expected values for a human when a human runs, how much energy they are using given a certain amount of body mass and a certain distance traveled. So it does seem to work quite well, at least in humans. Why am I telling you this? Well, if we've got a system that can predict how an animal moves and its maximum performance without any measured motion data, then in theory, we could use that system to predict how extinct animals move because we can't record their motion data, so we don't have it. But if we have a system that can predict it based purely on morphology, then maybe we can use this modeling framework to predict how dinosaurs or human ancestors or the earliest tetrapods to walk on land actually moved. And we can actually do this. We've been doing it for a decade or more. But what I want to talk about today is firstly why we would want to do that. And then for the bulk of the talk, how accurate are these reconstructions? This looks like a beautiful animation. Well, it's a simulation, not an animation, of a T-Rex walking along. But how accurate is it? How meaningful is it? And that's what I'm really going to talk about today. So I'll start with the why. Obviously, I hope I don't have to sell this as much to this particular audience as I do with some of the audiences that I present to. But basically, major transitions in the history of life involve biomechanics inherently. The transition between fish to the earliest land-dwelling tetrapods is fundamentally a biomechanical transition. There had to be modification to the limbs and the respiratory system in order for that animal to function on land compared to its sea-dwelling ancestors. Similarly, the evolution of flight in birds is fundamentally a biomechanical transition, as is the evolution of bipedalism in humans. And if we want to understand those transitions, then fossils are key. Fossils tell us the when, the where, and the how. And so being able to reconstruct the biomechanics of living organisms is fundamental to understanding how major ways of inhabiting the earth and major animal groups that inhabit the earth in different ways actually came to be. Now, traditionally, people wouldn't have used fancy simulation methods. They would have used skeletal proxies for biomechanical function. And I'm using the example here, the classic one of the evolution of bipedalism in humans and using the foot as kind of a hallmark for overall gait and locomotor habit, if you like, in our ancestors. Because modern extant arboreal apes have a fundamentally different size and shaped foot to the human foot, which has this stiff arch that supposedly provides this kind of like pole vault situation that makes our bipedalism more efficient, whereas the non-human ape foot is highly flexible for grasping branches. So traditionally, what people would have done is found a foot bone in the desert in Africa. They would have measured its size and shape, and they would have compared that size and shape to the same bones in modern apes. And if it came out more like a chimpanzee or an orangutan, they would have said, well, this human ancestor lived in the trees. Or if it was shaped more like a human bone, then they would have said, this is the first bipedal ancestor, and sent the paper off to nature and got promoted. Unfortunately, it's not that simple. And there are significant limitations to these more traditional approaches. Many of them are qualitative, quite often where there is a reasonable reason to suspect there would be a relationship between bone shape and mechanics. The relationship is sometimes variable or weak. And in fact, many of the relationships have never been tested, which was one of the reasons we tested the human example on one of the first slides that I showed earlier on and didn't find a relationship. Um, and then in some cases, there can't really be a skeletal proxy because the extinct group has no modern analog. And if I use the T-Rex example again, we don't have any 8,000 kilogram bipedal animals with massive heads and small arms alive today. So there is a real limit in that particular example in a T-Rex to how well you can take skeletal proxies and use them to infer how a T-Rex lived and moved. Hence, in the last 2 decades, people have moved more and more towards these biomechanical approaches where we actually use engineering software to simulate what these animals could or couldn't do because they're more quantitative. The limitation, of course, is that these techniques require information that is not preserved in the fossils. They require soft tissue information. Hence the question, how accurate can these reconstructions be? And that's what I want to explore today. And the basic way I think about this situation is to go really back to basics and go back to Isaac Newton. So if we want to understand or simulate how any animal, whether it be living or extinct, actually moved, so to understand its acceleration, we need to know something about the force that it can generate, its muscles, and the mass that those muscles have to move. So I've kind of symbolized this here in this diagram where the mass is represented by the body segment outlines, the kind of skin outline, if you like, that's the volume and mass of the animal. And then the force are our muscles here, that's simplified into lines of action. And the challenge, with all the first 6 slides that I showed today about the facilities that we have at Liverpool and the things that we can measure, with a living animal, we can measure all of these things and put the picture together. But with a fossil, these things are missing. We're missing those body segment geometrical properties and inertial properties. We're missing everything about the muscles. So how do we go about solving this equation when we don't have most of the information? And I'm going to go through how we do that and sort of evaluate where we are at as a discipline at doing a good job of this by looking at 2 case studies. So first, to look at mass, we're going to look at the evolution of body shape and locomotion in birds, where changes in body shape along the bird line were crucial to the evolution of not only flight, but also the way birds stand and move, their bipedalism. And then to look at how well we can reconstruct muscles in fossils, I'm going to use a case study that looks at the jaw muscles of rodents. So I'll go through these one at a time. So birds are extraordinarily diverse, both taxonomically, morphologically, and ecologically. And hopefully within these pictures, you can see that their body shape varies enormously as well. And that body shape went through a kind of, well, I guess approximately linear-ish trend during the evolution of birds, where at the top here you've got basal theropod or meat-eating carnivorous dinosaurs like T-Rex here and Allosaurus, which all have big heads, long muscular tails, and relatively short forelimbs. And as you go along this line to birds, and you get to, where's Archaeopteryx? Probably in here, Archaeopteryx. What you can see is that long muscular tail is getting shorter. The forelimbs are getting longer as they evolve into wings, and the head probably gets a little bit smaller as well. So there is this body shape change. And obviously flight has an awful lot to do with this. But what people have also noticed in birds is that birds stand and walk with a very unusual limb posture. They have this subhorizontal femur and very flexed, zigzagged limb postures. And that's very different to how humans stand and move. So humans stand much more upright like this with a center of mass over the foot and the limbs relatively straight. Whereas birds have this zigzag limb posture, which people have speculated is associated with having a center of mass that's further forward, so nearer the head. And so they need to have this zigzag posture to keep the ground reaction force over the foot so that they are statically stable. And it's possible that these changes were intrinsically linked. Basically, people think that basal theropod dinosaurs up here stood more like this, and this system gradually evolved along this line to birds as body shape changed. But we're not really sure what it has to do with flight and when body shape became like a modern bird, and therefore stance became like that of a modern bird. And if we can understand when that occurred, before or after flight, we can perhaps understand where flight evolved and whether this was mechanically necessary for flight to evolve. So how do we quantify center of mass position and body shape in our extinct dinosaurs? Well, traditionally, people did this very crudely. So they literally created what you might call scientifically constructed toys of dinosaurs. So these are hand-sized sculptures of dinosaurs, almost dinosaur toys, if you like. And of course, if you know the volume of your toy and you give it a density, then you get the mass, and you can then scale that up to the proportions of your real Stegosaurus. And if you dangle this from string and let it gradually stop rotating until it settles, it will tell you the center of mass of that animal as well. Now, this is obviously extremely crude. It's labor and skill intensive. You've got to be a very skilled artist to be able to do it. And it's probably not very repeatable and is definitely highly subjective. Things moved on, obviously, and when the advances in computer modeling techniques, people started to do exactly the same thing, but on a computer. So this is some work from a long time ago from my PhD, where I was digitally sculpting dinosaurs in order to estimate their body mass and center of mass. And in a later paper, we did this with a whole series of dinosaurs from basal theropods down here through to birds. So you can think of this as an upside down version of the family tree I showed on a few slides before. And by applying these models to all these animals, we could actually track or try to track center of mass evolution along this bird line. So the higher a dot is on this graph, the closer the center of mass is to the head. And there does seem to be this shift in center of mass away from the hip towards the head along the bird line. But of course, as I said, this is completely subjective. This is just me sculpting dinosaurs on a computer. And so in an attempt to be a little bit honest about it, we conduct sensitivity analyses and put error bars on our predictions. And what you can see is that back in 2013, if we were really honest about how certain we were in these trends, the error bars are absolutely enormous. And we could have absolutely no trend here whatsoever if we're honest about it, because it's entirely subjective. So how can we do better? How can we produce objective, empirically grounded estimates of body proportions and center of mass in fossils? Well, what we decided to do was to look at the living relatives of dinosaurs, to look at lizards, crocodiles, and birds, and to see if we could come up with a way to use their skeletons to better predict soft tissue volume purely from the skeleton. So essentially what we did was to generate 3D skeletons from CT scans and then use a mathematical algorithm, so an objective way, whoever presses the button gets the same answer, to kind of shrink wrap these skeletons with a volume. So you can think of this as kind of an external skeletal volume. So there's no room for muscle or anything like that. This is wrapped tight to the 3D bones. And then we also extracted the real skin volume from the CT scan. And we looked at the ratio between the skeletal, objectively derived skeletal volume and the real skin volume. And if you do that with enough animals and you plot this on your x-axis and this on your y-axis, what you actually have is a predictive relationship where you can predict the real skin volume with some 95% confidence intervals from information that's based purely on the bones. So we can take these relationships from the living relatives of dinosaurs and use them to predict the skin volumes, plus or minus whatever your 95% confidence intervals are, of extinct dinosaurs like T-Rex. And again, we've done that to a whole series of fossils across the bird line. And of course, this gives us a center of mass as well. And we get this really horrible graph, which I'm going to try and explain. So basically, this cartoon here illustrates what this graph shows. So here are the axes of this graph in black, with the red dot here is the hip joint. And then the data points are plotted on this graph. So the hip's up here, the head is somewhere over here, and the limbs are going straight down here to the floor. And what you can see, first of all, is that there is a separation in center of mass position between terrestrial and flying birds that makes perfect mechanical sense. Terrestrial birds that don't fly and have to walk efficiently on the ground have a center of mass closer to the hip than flying birds, which have a center of mass slightly closer to the shoulder, which is better for stable flight. Up here, we have a few lizards and crocs, and the bird data and the croc data is based on real skin outlines. And then we've used the method I described on the previous slide to predict center of mass positions for all these dinosaurs. And what you can see is that each dinosaur they actually have 4 data points, but often you can only see 3 of them because 2 of them are so similar, where we've reconstructed using different methods. So pretending that the dinosaur is purely a crocodile or a lizard, pretending it's purely a bird or an average of a bird and a crocodile. So we get different iterations. And you can see that these iterations don't make a huge amount of difference. So what do we see in terms of evolutionary patterns? Well, if we use modern crocs and lizards as an analog for dinosaur ancestors, their quadrupedal ancestors, then it would appear that the evolution of bipedalism in dinosaurs did exactly what you'd expect. It resulted in a backward shift in the center of mass towards the hip. What we then see is a strong phylogenetic trend along the line to birds, where in this region you have your stereotypical non-avian theropod dinosaurs. So your long tails, your big, scary heads with big teeth like T-Rex and Allosaurus are all up here. And then when things become much more bird-like, so things like your Velociraptor or your Microraptor that you might have seen in Jurassic Park, there is this downward shift in center of mass into the zone occupied by terrestrial birds. And then only with the evolution of flight in Archaeopteryx do we see dinosaurs or dinosaur-ish transitional forms entering the center of mass space that flying birds occupy. So we've got a really nice story here. We've probably got these 2 shifts in body shape and posture in the hind limb along the line to birds that suggests perhaps that there was some change in bipedalism going on before the evolution of flight. And it was only after evolution of flight that we see this fully flexed bird way of standing and moving on the ground. That's a nice story, but how does it stand up? How accurate are these predictions likely to be of the dinosaurs? Well, one thing we can do to try and evaluate that is basically pretend that our living animals in this sample are fossils and apply these predictive techniques to our living animals to see how well they work. And that's what we've done here with the birds. So the blue zone and the red zone here are the skin volume morphospaces from this plot I've just explained. And then the new data points and the convex hulls around them in red and blue are the same birds, but we've applied the predictive techniques to their skeletons, pretended they were fossils. And in fact, we've pretended that they are an average of a bird and a crocodile. So we've not even been fair to ourselves and said, well, these are obviously bird-like, we should use a bird prediction. We filtered in croc and lizard data as well to see how wrong our answer becomes. And as you can see, it doesn't become very wrong at all. This blue convex hull here is very similar to this blue shaded region, as is this red convex hull and data points to this red shaded region. And the qualitative differences, obviously, because the quantitative differences are so small, the qualitative picture doesn't change at all. So that's a really nice result in terms of some confidence in our new objective method. The other subtle point I'll make is that obviously this is slightly circular because we're using the inputs into the predictive equation as a measure of accuracy. If we apply two birds that are not in the data set here um, through this process, we get very similar results as well. So it's likely that this works across birds more generally. So for mass, we can give ourselves a bit of a tick. We've got an objective method that seems to work. What about force? What about the muscles? So for this, I'm going to talk briefly about the evolution of mastication in rodents, because rodents are a really nice case study to look at muscles, because their jaw muscles are extremely definitive of the way that they feed and the things that they feed on. And people have, for a very long time, split rodents up into three morphotypes that are kind of phylogenetically independent, if you like, or convergent. So these morphotypes evolve repeatedly across rodents and are associated with dietary shifts. Um, and it's really all about the different relative sizes of these muscle groups that I'm not going to go into and name because we don't need to get into that. And these different muscle proportions seem to be highly predictive of the way that these animals eat. So there are links with maximum bite force and efficiency and stress magnitudes and stress distribution across the skull as these animals feed. So for example, in our squirrel and guinea pig type, I'm not going to try and pronounce these words. I'll just use the squirrel and guinea pig type terms. We see that the squirrel type morphology is more efficient at incisor gnawing than the guinea pig type, which is more efficient at processing food with the molars, which is entirely consistent with how these animals feed. Whereas our rat type morphology seems to be adaptive for high performance generalism, so being mechanically efficient at all bites. So what we wanted to do in this situation was essentially what I've just done with the birds, is throw that real muscle data away that we've already measured and pretend these rodents, these guinea pigs, rats and squirrels were fossils and apply the muscle reconstruction methods to them as if they were fossils, and then compare our predictions to the real measurements of jaw muscle size. So we did this. We tested two of the most common methods for reconstructing how big muscles are in fossils and how much force they can produce. The first was sculpting the muscles in a CAD package in a similar way to I showed before about reconstructing dinosaur body mass. And then also to use different ways of estimating how big a muscle is based on its attachment area or in a particular zone of the skull that you can define objectively. So we did this in a couple of different ways. We had three investigators subjectively sculpt muscles around our 3D models of rats, guinea pigs, and squirrels. And then we also measured muscle attachment areas in the skulls and plugged those numbers into equations that predict how big the muscles are and compared those values to the real muscle sizes in these animals. What we found is an awful lot of results, which I cannot possibly go into here. I've referenced the papers here in case anyone's interested, so I'm just going to do a horribly quick summary of the results. So in terms of quantitative accuracy, what we found was that volume sculpture, so these volumes that people sculpted to mimic the real muscles, were highly variable. Two of the investigators that did it got average errors of 70% and 94% across their muscles. One of them was much better and averaged 12% error. And then in terms of our attachment area methods, which aim to predict how big the muscles are based on areas of the skull, one set of methods produced an average error between 25 and 40%. But in some cases, the errors across individual muscles were absolutely huge and up at like 300%. So highly variable again. Now, a paleontologist might turn around and say, well, you'd expect them to be quantitatively inaccurate because these are crude methods. But what a paleontologist would hope is that the relative differences between animals is correct. So if a muscle is bigger in a rat than a guinea pig, the method predicts that relative difference correctly. So is that actually what we found? Unfortunately, in many cases, it's not. So with the volume sculpture, in one case, the best case scenario, which was our investigator two, who was more accurate quantitatively, They only got 75% of them ordered correctly. Worst case scenario, only a third of them had relative accuracy predicted correctly in terms of how big the muscles were across the three species. And our attachment areas really weren't much better. 1 out of 6 correct in one particular method, and then in a different attachment area method, 40% of them correct. So not really very encouraging overall. So if we go back to our big picture, while we've done quite well reconstructing mass properties and body shape in fossils, there is this question mark about how well we can actually reconstruct muscles and therefore the kind of muscle forces that an animal could produce, which is obviously highly determinate of how fast they could run, how hard they could bite, etc. So where does that leave us? I've shown you a really pretty animation of a T-Rex running along, but if we can only reconstruct its mass properties well and its muscle properties not so well, how does that leave us, reconstructing its overall movements and then other sorts of functional transitions in the fossil record? Well, to sort of look at the current state of affairs and the way that we have to approach that currently, given these limitations, I'm going to run through a study that we've just published where we attempted to predict running performance in Australopithecus afarensis. So I'll start with the why again. Why would we want to look at running performance in Australopithecus afarensis? Well, bipedalism is obviously a defining feature in our own evolutionary history. And the kind of, well, I don't know if transition is the right word, but certainly the differences between Australopithecines and our own genus Homo have been a focal point for this research. And that's partly because we've got near complete skeletons like the Lucy fossil, but also we've got other good evidence like fossil footprints that can tell us about bipedalism in these animals. But also we've got major changes in anatomy or major differences in anatomy between these animals, and particularly changing body proportions, which would have obvious implications for locomotion. And perhaps because of the footprint evidence, most of the research has focused on how Australopithecus afarensis walked. And although there's been arguments through the years, the consensus in recent times, based on the footprints and based on the kind of biomechanical simulations that I've been showing you, is that Australopithecus afarensis walked relatively upright in a human-ish kind of way with relatively low energy costs. And that's kind of fossil footprints, simulations, and when you put the footprints and the simulations together are all pointing in that direction. And that's really cool and interesting, but it raises the question, obviously, that if Australopithecus was already walking relatively upright and relatively economically, why these major changes in body proportions at genus Homo? If walking was already highly, well, maybe highly is not the right word, but certainly well adapted to walking, why did they change? And the obvious thing to look at is running. Maybe that's the solution. But to date, at least before our study, there had not been any attempt to quantitatively estimate running performance in Australopithecus afarensis. So if we go through our process again, this is what we did. We used the same sort of objective shape fitting and shape expansion technique that we used in the dinosaurs on Australopithecus, but obviously we used a mammalian data set to expand the volumes rather than a croc lizard bird data set. Don't know why it keeps getting stuck. And then we used an attachment area method similar to the rodents to predict the sizes of the muscles. Now, obviously, I've just said that that's a highly limited approach. So what we did, as it happened, the prediction that came out was almost exactly average between a human and a chimpanzee. So what we did was to not just run one model, but we ran models where, well, two extra models, one where leg muscle mass was reduced to the percentage body mass seen in a chimpanzee, and one where it was increased to the percentage body mass we see in a human. And we also did similar things with other key aspects of anatomy, like the architecture of the ankle extensor muscles down here and the Achilles tendon. And then something I'm not going to talk about today because there isn't time, we also looked at how contraction velocity, which is also expected to differ between arboreal and terrestrial apes, affected the model prediction. So we basically ran lots and lots of different models to try and understand maximum and minimum limits on running performance, but also how these traits, which evolved from one state to the other at some point, would have affected running performance by themselves. And what we did was something similar to what I showed in the jumping animations, where having defined the model's anatomy, you ask it to search for different muscle activation patterns that result in the fastest and most economical running gait. And it starts off, of course, by falling straight on the floor, but gradually it gets better. If I can make the slide change, gradually it gets better and starts to find something which is clumsy and manages a couple of meters in a very uneconomical and unattractive way before it falls flat on its face. And then after even more and more iterations of learning and trying different activation patterns, it's still a bit silly, but now it's much more stable and it can manage continuous locomotion over quite a long distance. And then if we keep going, we get fully evolved running gaits. And these are two example reconstructions that highlight one of our major findings. So these models both have the same mass in their limbs that we predicted as a sort of average between human and chimpanzees. But this faster model here, which is also using less energy, has a human ankle muscle and Achilles tendon, whereas this one has a fairly extreme non-human ape arboreal ankle muscle architecture and Achilles tendon. And what you can see down here, if you look at the numbers, is that the human one is twice as fast almost and uses about 50% less energy to achieve that faster speed. But in any case, even if we make the muscles as big as a modern human and give the model modern human-like ankle properties, it still only manages 5 meters per second. And we predicted that the walk to run transition occurred at 1.5 meters per second. So we've got a very narrow running speed range here, which I'll come back to in a few slides' time. In terms of energetics, we also found that the energetics of the model was highly dependent on the ankle muscle architecture. So if we gave it human-like ankle muscles, which are these solid data points in here, Lucy's predicted to be as economical as a modern bird or mammal. It's within this grey zone here that you would predict for an animal of Lucy's body size. But if we take away the human-like adaptations in the ankle, you can see that these open data points are all above this grey zone. So without that adaptation in ankle muscle architecture, Lucy would be more expensive or less economical than a modern mammal or bird of its body size, but still more economical than a chimpanzee, whether that chimpanzee is walking bipedally or quadrupedally. So in summary of that study, even with the limitations surrounding soft tissue properties and muscles in fossils and predicting those accurately, we can be fairly confident that Australopithecus' top speed was lower in absolute or relative terms than modern humans. We've also shown that how we reconstruct the ankle musculature makes a huge difference to both top speed and energetic cost. And coming back to that idea of a contracted running speed range, it means that the range of running speeds that are available for jogging or sub-maximal effort running was much lower in Australopithecus than it is in modern humans. Even more so if the lower muscle masses or the ankle extensions were more like non-human apes. So the amount of endurance running or the capacity for endurance running in Australopithecus, we can make an indirect argument that was probably much more reduced or lesser than modern humans. But overall, even though we can't accurately predict how fast Australopithecus could run and how much energy she used, it does seem that there was a jump in running performance at the evolution of Homo, and that perhaps the human body plan was more about running than it was walking. So in terms of overall conclusions for the entire talk, I think what I want to everyone to take away from this is that there is this inevitability of missing data when we start looking at fossils. But there is hope when it comes to reconstructing the biomechanics of major evolutionary transitions despite that missing data. And the biggest successes have come where we've actually measured anatomy and function in living animals first before going to the fossils and developed new ways to link bony morphology to soft tissue in living animals that can then be applied to fossils. But of course, there will be a limit to what we can understand, but we're not yet there yet. There's still more work to be done and still improvements to be made. But there will be some things that forever defy explanation. And that's where I'd like to stop. So thank you very much for listening. And thank you to all these people for their help with this work and the funders as well.
Thank you very much. That was absolutely fascinating.
Sorry, I've gone over a bit.
No, no, don't worry. We started 2 minutes late. So that's fine. If you want to unshare your screen and then we can be in a gallery and have a look at each other for. Um, yeah, that was really fascinating. And thanks for also showing the kind of facilities that we have at the university. It's really, really cool to see what you're doing, and really enjoyed it. So as always, if anybody has any questions, you can either raise your hand and I will say your name and you can ask a question out loud, or feel free to put questions in the chat and I will read them out loud for you. So we've got a hand up from Walter, if you'd like to ask a question.
Sure. Thank you for calling on me. This was a very interesting and thought-provoking lecture and very well done research. I want to congratulate you on that. It's also a very nice and healthy use of artificial intelligence, as opposed to, a poor use of it. So I'm interested, has anyone done any work on evolution of the hand? And also, a second question, if it's possible, has anyone done any work on the evolution of the size of the posterior fossa, trying to get an assessment of the cerebellar size, maybe cerebellar anatomy by internal skull impressions, as well as the occipital area for vision, and the posterior frontal area for locomotion. I don't know if I made myself clear.
Yeah. So the first thing I will say is that I'm not an anthropologist at all.
Okay.
So I knew my phone would ring. I'll ignore it. Um, in terms of the hand, the person's research to look at would be Tracy Cavell, who is at the Max Planck in Germany. I'm not incredibly familiar with it, but I know that she's done a lot of similar stuff methodologically to the stuff I presented today, and she is the big name in that area. And she's actually used our X-ray facility to look at tool use and how the carpal bones move as we use different tools. In terms of your second question, I have absolutely no idea. Sorry.
Right. How do you spell Cavell?
Kivell.
All right, thank you.
Sorry, I can't be more helpful.
That's quite all right. You've said a lot. Good reference.
Thanks very much. I wondered, after you had done the more recent studies on maths, did you compare back to your original ones that you'd done as part of your PhD, and did you ever work out how accurate you were with your subjective ideas?
Um, well, yes.
You don't have to answer if you don't want to.
I mean, in many cases, I think what comes out of the more recent study more than anything is that the birds are different, and no one had ever considered that at all. And what we found was that there wasn't this dinosaurs have a center of mass near the hip, birds have a center of mass further forwards. We found that terrestrial birds and dinosaurs overlapped with each other, and then anything that flies has that further forward center of mass. So qualitatively, that was a big shift in thinking. But the actual predictions for individual dinosaurs mostly overlapped. But obviously we've got this objective way of doing it now. I think where the shift occurred also um, was slightly different in that previously we'd got a more gradual shift to the bird-like state, whereas with the new results, even things like Velociraptor are still much closer to the hip. And then there's this big jump at Archaeopteryx.
Mhm.
With those two exceptions, Allosaurus and T-Rex were pretty much the same, but there was those other differences.
Oh, cool. Thanks. It's yeah, really cool study. And we've got a couple of people in the chat who have not asked questions, but we've had several people. So Gabrielle, Isabel, and Amy all said, thank you very much. And you've answered many of my labelman questions about how information is extrapolated from fossils. That's from Isabel. And from Amy saying, I'd heard various bits of your research before, but it was really nice to see how the whole picture and everything fits together, which, yeah, great comment.
Thank you.
If anybody else has any questions, please feel free to write them in the chat and or ask out loud.
I've got a question about the seminar series, if that's all right.
Yeah, of course.
Am I a bit of an odd one out by presenting stuff that isn't completely anthropology, or do you often get non-anthropology talks?
We have a range. I think I would say that we mostly have anthropology, but we have had a few. So we had one a few years ago that was about robotics, and kind of looking to the future rather than the past. And so last week was about working memory, and that was more psychology than like strictly evolutionary anthropology. So we do go out into other disciplines as well. Yeah.
Cool.
Yeah, it's one of the nice things about evolutionary anthropology is that you can take it in. There's quite a lot of overlapping disciplines.
Yeah. So
Um,
which department are you based in then?
Archaeology, classics, and Egyptology.
Okay.
hmm Um, yeah, the seminar series has been running since as long as I was an undergrad. I think it was started in 2017 or so. But it really took off online over COVID.
Right.
hmm Oh, we have a question from Anthony in the chat. Saying, Hi, Karl. I've noticed that you've looked at the differences in efficiency in relation to the surface that movement happens. Would there be a difference if considering different temperatures or climates?
Quite probably, yeah. So I mean, one of the if you go back to Dave Carrier's work on endurance running from the 1980s, he talks about um, yeah, about temperature and climate and how that probably influenced um, our capacity to run long distances, the importance of sweating, for example. So yeah, absolutely. And we did have a grant in to look at that quite a long time ago now that didn't get funded, but yeah. So yeah, it absolutely would.
Thanks, Anthony, and thanks, Karl. Do you have any ideas of what it would take to improve the force models that you were making? I know obviously in your study you kind of got around that problem by making lots of different simulations. But do you think that there will be in the future an objective method in the same way that there is for mass?
We are in the process of writing a paper. So
Oh,
if we give
exciting.
this talk a year from now, it would hopefully have two ticks, not a tick and a question mark. But we've only done it for birds and crocodiles. So the same data set that we've used for the body mass stuff, we've used to develop new attachment area equations and also some other equations based on those convex hulls, those shrink wrapped volumes where we use them again as our x-axis and we have muscle mass on our y-axis, and so you can predict one from the other. And the prediction intervals that we get, so confidence intervals, if you like, from that method is miles better than anything that's been out there before. So we're now at plus or minus 20% for muscles in birds and crocs and lizards and therefore dinosaurs by inference. Um, yeah, so we're in the process of writing that up now. The problem with and I did exactly the same thing in the Lucy paper, but the problem there is that you'd have to go to all mammals. I did it for non-human apes and gibbons, which gives you an N of 5 or 6, I think. And your regression model is never going to be strong with an N of 5 or 6. And so the prediction intervals, confidence intervals on that were enormous, plus or minus 300% or something, because you've only got an N of 6. Whereas with birds and crocs, we've got thousands of species. Birds, crocs, lizards, yeah, thousands of species. So we can have a huge N and produce nice predictive models.
Yeah, I see. I see. That sounds exciting, though, for the birds and crocs at least.
Yeah, and we got some nice results applying it to dinosaurs as well in terms of, when flight might have evolved and when this weird zigzag posture might have evolved as well. So, yeah.
We have a very nice comment from Isabel, just coming back to our chatting about the series. Thank you very much, Isabel. It's really nice to be able to put it on. And then we have a comment from John as well, just relating to your talk. It's really good to hear this. And he had to miss most of it, but from the end, he says you're very much within paleoanthropology for us.
Oh, I
So
don't know about that.
the Lucy paper.
Yeah.
If we've got no more questions, I can't see any in the chat. I think we will wrap up because we've just gone to 2 o'clock. But thank you for such a fascinating talk, and good luck with all of your upcoming papers. Everything sounds really cool, and I have really, really enjoyed listening.
No, you're welcome. Thank you very much for hosting me.
Thanks.
Bye bye.
And thanks for joining, everyone. Goodbye.